Okay, great. Hello, everybody. I'm Christine Escobar, and I'm very excited for this opportunity to tell you about the lunar effects on agricultural flora experiment, or LEAF. It's an instrument being developed for Artemis 3 deployment. And I'm really proud to count myself amongst a group of renowned and talented space biologists and engineers, which includes our co-investigators from our company, Space Lab in Boulder, Colorado, biologists from the University of Colorado, as well as from Purdue University. And we also have two co-Is from Australia, from the University of Adelaide and La Trobe University, as well as space biologists from the NASA Kennedy Space Center. So let's start with a question. Why do we want to grow plants on a spacecraft? Because living in space is a tremendous sustainability challenge. The farther we travel from Earth, the more it pays off to reuse and recycle what you consume, rather than carrying all of your supplies with you and throwing away the waste. And while packaged food stores have limited shelf lives, they lose quality over time, plants can provide us a regenerable, nutritious food source. They turn our carbon dioxide into oxygen, they can purify wastewater, and they can make us happy and reduce our stress. Plants can also produce many products that we use in our daily lives, like medicine and fuel and clothing. So human nutrition and life support provided by plants can enable long-duration human exploration of the moon and beyond. And that requires an understanding of the space environment effects on crop physiology and growth. So in space, we need all the same things to grow a plant, right? You need water, CO2, minerals, and light. But there's a lot of complications. You're extremely limited by how much mass you can carry by available volume and power. Microgravity, or in the moon, reduced gravity, complicates water transport, complicates air mixing and root aeration, and it confuses the response. You've also got high radiation levels, reduced pressures inside human cabins, spacecraft cabins, sometimes elevated carbon dioxide, and even high acceleration loads, which all these things can physiologically stress the plant and the hardware that's supporting them. It's even harder to control infection or buffer chemical releases in a small, isolated space container. And there's something you can't go to the store and buy a new part. So through decades of research in ground-based test facilities, we've made great strides in controlled environment agriculture in space and us. And this has paved the way for operational space farming. There's also been many plant growth studies conducted in low Earth orbit with small growth chambers. We've learned a lot about the effects of the space environment, especially microgravity, on seed germination, on plant growth, on morphology, under stress, on how they take up water and nutrients, and gas exchange. We measured microbial loads in plant material. And we've even peered into the plant genome to see how the space environment affects gene expression and molecular physiology. We've demonstrated some great enabling technologies that we can use in microgravity, like slow-release fertilizer plants, seed films, and capillary-driven water transport. And now, in order to prepare for our sustained lunar presence, we'll need to continue such research on the moon to assess the potential for sustained crop production. So LEAP's primary science goal is to understand the effects of that lunar surface environment, 1/6 gravity, elevated radiation, in a cabin-like atmosphere on model space crops at an organism-wide scale. Our first objective is to grow model space crops that we think will have stress resilience and grow them in the lunar and Earth environment. And then second, we'll compare crop phenotypes in those lunar and Earth controlled environments, which includes looking at seed germination, clonal reproduction rates, changes in plant morphology, their size, their orientation, their color, the plant's growth rates, and their photosynthesis respiration rates. And then finally, we aim to identify genome-wide biomolecular deviations of the lunar and Earth exposed crops. And we really hope that this LEAF experiment will provide a foundation for future studies of long-term multi-generational spacecraft performance. So to accomplish these objectives, we have the LEAF payload shown here conceptually, and basically it's a growth chamber that provides the plants within with the light and the nutrients and the water and the atmosphere that they need to grow, while it protects them from excessive lunar sunlight, from radiation of space. The LEAF is a Pathfinder experiment to help us predict spacecraft effectiveness as life support components that provide a source of nutrition while they convert our... Okay, apologies for the pause. So again, we hope that LEAF will be a Pathfinder experiment that will help us predict how effective our spacecrafts will be as life support components that provide us nutrition while converting our respired carbon dioxide back into breathable oxygen. So this experiment, what we want to do is link the biophysical stress that's imposed by that environment on the moon to how regulatory gene networks and organism-wide function shifts in response to that stress. And these reactions or responses might afford plants certain stress resilience. Those fundamental relationships, then we can use that to model and predict the crop's long-term potential for germination and growth, for nutritional quality, and for photosynthetic gas exchange in deep space. And then this knowledge hopefully can then be used to select more space-worthy crops, even enhance them via breeding or genetic engineering, and also will help us to design equipment to provide the most optimal growth conditions for our plants to thrive. What do we expect to see? Ionizing radiation is a key biophysical challenge in human space exploration because it produces reactive oxygen species, which can damage biomolecules, causing a variety of detrimental effects. Antioxidants are the first responders of reactive oxygen species, or ROS formation, and spacecrafts can provide our humans with a dietary source of those antioxidants. Our plant's redox state describes the balance between those oxidants and antioxidants and serves as a regulator of plant metabolism, of germination and growth, of their development, and even their defenses. So we expect that space radiation and reduced gravity on the moon will cause overproduction of oxidants and then shift the plant's redox state. And then what we're really excited about is by returning our plant samples to Earth, we can then observe those shifts by looking at changes in plant gene expression when they're exposed to the lunar environment. And this will give us insight into genetic indicators for long-term crop resilience and quality, such as having a higher capacity to produce antioxidants. LEAF will house 3 model plants that have been studied in space. 2 of those are representative of potential crops that could produce edible, nutritious biomass. This includes green and red varieties of fast-growing brassica rapa, specifically Wisconsin fast plants. These are in the same family as mustard plants and cabbage. We'll have floating aquatic plants called wolffia, also known as duckweed, or watermeal. And these little plants are incredibly fast-growing and nutritionally dense. Duckweeds are not only well-known model plants, but they're also they were the first plants used to observe photosynthesis in space. And then finally, we will be growing Arabidopsis thaliana, which is a model plant with extensive spaceflight heritage. And as a result, there's extensive data sets in NASA archives, which can inform our studies. The LEAF experiment includes a flight unit and a ground analog unit. Each are going to have isolated atmosphere. The 2 twin growth chambers will replicate typical cabin air with elevated carbon dioxide that would normally be respired by crew and a reduced pressure of 8.2 psi. LEAF instrumentation includes acceleration monitors, radiation dose monitors, a camera for capturing multispectral images of the seedlings, and sensors to monitor the plant atmosphere, specifically its temperature, pressure, and gas composition. The plant habitat will support their growth with a supportive substrate, a reservoir of water and dissolved nutrients with LED lighting, and a preloaded sealed atmosphere. In our pre-deployment phase, the payload is going to maintain conditions for seed inactivity. Basically, the payload is off and inactive during HLS launch, lunar transit, loiter in the NRHO or near rectilinear halo orbit. They'll be inactive during crew arrival and docking in the Orion capsule, and then during HLS landing on the lunar surface. And then in the deployment phase, the Artemis crew will unmount the payload from the HLS, egress from the vehicle, carry it and its base to its deployment site, place it on a level surface, verify its alignment with the gravity vector with a bubble level, preferably within 10 degrees, and then rotate the payload so that the solar panels are aligned with the lunar east-west axis with the sun, using a sundial, much like that used on Apollo instruments. And then in our initialization phase, the crew will unfold solar panels, which will allow LEAF to power up. Autonomous self-checks and instrument calibrations will proceed while LEAF transmits its status to a ground station on Earth. And once our ground station operators review and confirm payload power and system function, they'll send a command back to LEAF to commence the experiment. There'll be LED lights on the payload structure, which will indicate readiness for experiment activation to the Artemis crew. And then the crew will water the seeds inside by depressing a plunger on the LEAF structure using a special tool that's designed for their spacesuit gloves. The crew will then take a picture of LEAF before they commence on the rest of their Artemis EVA activities. And then during our surface phase, the code will control conditions for seed germination and growth while it collects imagery of the seedlings, monitors their gas exchange, and monitors both lunar gravity and radiation inside the chamber. Payload monitoring and environmental control will continue for the remainder of the lunar day, even after the crew leaves, until solar power is lost. And then before the crew departs, before Artemis crew departs the lunar surface for the return to Earth, after about 3 to 5 days on the surface, they'll come back to the LEAF payload to fix and retrieve a portion of the growing seedlings. And then those fixed seedling samples will travel back with the crew to the HLS, to the Orion capsule, and back to Earth. And finally, the LEAF science team will analyze the returned fixed samples of plant tissue. First, we're going to have NASA Gene Lab helping us to conduct bulk transcriptomics using their well-established protocols. And then we'll have some debris from that analysis that will be recovered at Gene Lab and sent to the University of Adelaide to assess cell wall structure. We'll have La Trobe University analyze single-cell gene expression for a portion of those returned samples. And this will help us observe how different types of plant cells respond. If successful, this will be the first application of single-cell transcriptomics to plant gravitational biology. Biologists at the Kennedy Space Center will characterize the microbiome present in the plant tissue. And if there's enough material left, we can identify and quantify proteins as well. So in summary, this opportunity is very unique to be able to fix and return plant samples to Earth. And it allows us a systems biology approach to identify stress-induced shifts in the plant's genetic expression, in their biochemistry, and even in their microbiome. We'll be conducting regulatory network modeling, which basically that will link those shifts in observable changes in plant phenotype or function to their shifts in their biomolecular and genetic expression. So in other words, by comparing the lunar-grown plant samples to the ones grown in the ground control unit, we can attempt to define a lunar environment transcriptomics signature, and that hopefully we can use to predict the productivity and nutritional quality of potential lunar spacecrafts. So we're really excited to embark on this amazing 3-year journey, and we can't wait to share with you our results in the future. Thank you.